Good evening. Firstly, I would like to thank the scientific committee and the IAPRD for inviting me to give this talk as part of this educational session entitled Translational Treatment in Parkinson's Disease. My topic is pharmacotherapy for PD, and for the reasons I will tell you now, I have added in the title, What's in the Horizon? So here you can see my disclosures. And without more preambles, I would like to start. So this is the invite letter I received to give this lecture, which obviously I was delighted to receive and to accept. And then I started thinking, because this is an educational session, so I presume that part of the audience might be trainees and residents, but also this is a congress for specialists in movement disorders. So the session was entitled Translational Treatment in Parkinson's Disease, so I wanted to go a bit deeper in the topic rather than just telling the current and conventional treatments that we are using in daily practice. So that's the reason why the outline of the talk is just a very brief reminder of what is the current pharmacotherapy in Parkinson's disease, both for treating the symptoms and also for handling the chronic treatment complications. And then I would like to expand a bit more in research in pharmacotherapy. So what's in the horizon, as I was telling you in the title of the talk. So, and maybe one of the major issues in this arena is moving towards disease modification rather than just treating the symptoms. So I will briefly just overview the methods to define disease modification, which is a really very difficult topic and would deserve a talk on its own, right? Then the potential targets that are under investigation, like cell therapy and alpha-synuclein directed therapies, but also other pathways like insulin, GBA, iron, and then the possibility of in the future having personalized treatments. And also, very briefly again, just a few words on the treatment of severe non-motor symptoms, for which the main example could be dementia. And then I will try to end up with a summary for this talk. So what are the current main pharmacotherapy approaches used in the clinic to treat Parkinson's disease? And obviously there are strategies in this slide that are not actually pharmacotherapeutic, but I just wanted to give a very general overview of what we do nowadays to treat Parkinson's disease. So obviously, as you all will know, we have the levodopa and the dopa decarboxylase inhibitors. We have all the dopamine agonists, oral, subcutaneous, transdermal. We have the enzyme inhibitors. We have amantadine and anticholinergics. We have drug infusions, which can be either levodopa or one of the dopamine agonists, which is apomorphine. Then we have deep brain stimulation. As I was saying, this is not a properly pharmacotherapeutical approach. And then we have other treatments like botulinum toxin, cholinesterase inhibitors, physiotherapy, which again would not be a properly physiotherapeutic approach, laxatives, antidepressants, and much more. So as you all know, to treat motor symptoms in Parkinson's disease, we have a lot of strategies, and this slide tries just to give a general overview on what we do during disease progression if we think about a particular patient. So at the beginning, we have the diagnosis and the initial treatments with what has been called the honeymoon or the good years. So in these first years, usually for the most part of the patients, we can control to a very satisfactory extent the motor symptoms of the disease using oral conventional treatment, be it dopamine agonists, enzyme inhibitors, or levodopa itself. But then throughout the years, there can appear this progression of symptoms, and also these chronic complications of the treatment, mostly levodopa, and that include motor fluctuations, but also dyskinesias, and obviously other side effects of other treatments. We have been worried for the last decades a lot about the impulse control disorders, for instance, in the case mostly of the dopamine agonists. So throughout all these years, we have at the beginning, in these good years, the oral conventional treatments. Then when the symptoms increase and there are very early motor complications, we can start adding some more drugs like enzyme inhibitors, and also start using apomorphine, for instance. And then when apomorphine, like in Penject, I mean, and then when the motor complications get worse and much more complicated and difficult to handle with conventional treatments, then we can use the drug infusions, be it apomorphine or levodopa intestinal gel, and then obviously deep brain stimulation. So this would be an overview of more or less what we all do in clinical practice. And here you have pictures you all know. So of apomorphine, you can use it like acute challenge or Penjects. So this can help the patients to overcome an acute severe off period. And that's what you see on the left-hand side of the slide. And then on the right-hand side, you can see the pump, the subcutaneous continuous infusion of apomorphine that can help patients to control motor fluctuations as a continuous infusion, as I was saying. Another approach that I have also mentioned is duodenal infusion of levodopa, that you also know, and that is approved and used in many countries. Usually what we do is to have the patient admitted in the ward, or sometimes we can do it in the outpatient. So a test period with a nasoduodenal tube, and just to test and check if the patient has a good response to the duodenal infusion of levodopa, and then the surgery of endoscopical treatment, which is placing the duodenal tube through the abdominal wall and connecting it to the pump for the chronic treatment to help us handle the motor complications of Parkinson's disease. And then obviously, as even though it's not a pharmacotherapy, and this is the topic of the speaker who follows me, but we have deep brain stimulation, who has been used for several decades now, and which has very high class evidence of its usefulness to handle motor complications. I am not getting into the details. And the same with HIFU, that I think also is part of the lecture of the next speaker. But just because, even though it's an educational session, I think that many of us are very familiar with the daily use of the conventional treatments, I wanted to give you my take on what are the main pharmacotherapy research lines in Parkinson's disease at present, and what could be in the horizon, what are the caveats and the concerns, and what do we have to think about when trying to be critical about what we are doing, and if we can learn from the clinical trials that end up being negative, for instance. So I think that the main pharmacotherapy research lines in PD at present, and what's in the horizon, as I was saying in the title, still do include dopaminergic replacement strategies. We are still conducting clinical trials on this, even though it's a symptomatic approach. But then we have all these new approaches that have several ongoing and planned clinical trials that try to move forward from symptomatic treatment towards disease modification. So, as I was saying in the outline, we have alpha-synuclein related strategies. Then we have strategies targeting genes that have been related to familial, but also in some instances to sporadic Parkinson's disease, like LAC2 gene. Then we have GBA, which is an important genetic risk factor for Parkinson's disease and Lewy body disorders. And then we have other, like insulin and iron, that for several decades have attracted attention and have clinical trials underway or planned as well. So just as an example that we are still working with new approaches for dopaminergic replacement to treat the symptoms of Parkinson's disease. Last year, the results of the clinical trial, a phase 3 study on apomorphine sublingual film was published, as you all know, and you can see in this slide. So you can see the flow chart of the study. So in the end, like 54 participants were assigned to apomorphine sublingual film and 55 to placebo. And there were some withdrawals, but then in the modified intention to treat population, all the patients were assessed. So just very briefly as well, in this table you can see that as expected, the scales that capture the motor symptoms like the UPDRS-3 and also the response in terms of the time to reach an on when starting from the off period were significantly better in the apomorphine sublingual film group, the experimental group, compared to placebo. And other measures like quality of life or other scales were not that significantly improved, but the scales or the variables that captured the response of the motor symptoms were significantly better as expected in the apomorphine sublingual group compared to placebo. And here you can see the same information in terms of the change in the MDS UPDRS para 3 score. So this improved more in the apomorphine sublingual film. And then the transient adverse events were more frequent in the apomorphine sublingual film, but still the therapy was relatively well tolerated. Then we have the cell-based therapies that not all of them, but a lot of them are also focused in dopamine replacement. So part of these cell-based therapies trials have focused on placing cells that can produce dopamine in the striatum. So these therapies have the problem, obviously, as other approaches, of being invasive. And they have side effects related to the graft, like this was reported several decades ago with the first trials like graft-induced dyskinesias. But also we could have the concern of the side effects related to the host, so maybe the disease propagation to the graft, even though it has been shown not to happen in all the cells that are grafted, but in a percentage of them, and it can take several years to happen. But still, this is a concern related to this prion-like hypothesis of Parkinson's disease, to which I will get back later on. So, as I was saying in the introduction, it is really difficult to define what is disease-modifying effect of a therapy when we perform a clinical trial. And in this review article by Olano and colleagues, this topic is reviewed. So there are several endpoints that have been used, and this is a reflection that none of them is perfect, probably, because otherwise we would be always using the same endpoint. So time to need levodopa, the increase in UPDRS, then dopaminergic imaging, and also the early delayed start paradigms have been used to try and separate the possible symptomatic effect of a given intervention from its potential disease-modifying effect, if any. So in this figure, which is a bit busy, but just to highlight some parts of the degenerative cascade that have been proposed to be relevant in Parkinson's disease. So, like the LRRK2 gene I was mentioning before, that could be important not only in Parkinson's cases that are carriers of a mutation, but maybe it could be relevant also in sporadic Parkinson's disease. The same with iron, that for decades it has been an ongoing controversy as to whether or not iron is an epiphenomenon or is really part of the pathophysiological cascade in Parkinson's disease. Then we have oxidative stress, inflammation, obviously the glucocerebrosidase and the ceramide way that has been implied and related also to alpha-synuclein itself. So all these, the mitochondrial dysfunction, and as we will mention later on as well, the role of insulin, all these players have been proposed to be relevant in the pathophysiology of Parkinson's disease, and subsequently have been also proposed to be potential targets for treating the disease and trying to modify the disease evolution. In this review that I recommend, the topic of antibodies for treating Parkinson's disease is overviewed, and I think this figure is nice. It's showing that there are several points where the use of antibodies against alpha-synuclein in any of its forms could potentially be helpful in the degenerative cascade in Parkinson's disease. So here you can see this is also a very busy figure. But for instance, the antibodies could target oligomeric or fibular alpha-synuclein, or they could create immune complexes that would be internalized by microglia for degradation, or there could be also targeting of cell surface receptors to prevent entry of alpha-synuclein into the cell, or there could be intrabodies that would bind intracellular alpha-synuclein and stimulate its degradation. So there are several hypothetical or theoretical points of the cascade where antibodies could be helpful, but obviously this needs to be demonstrated. Here we have active immunization against alpha-synuclein. So here we could use oligomers of alpha-synuclein to induce the generation by the body itself of antibodies that would be the ones that would try to clear or to stop some of the points I was referring to in the previous slide to modify the disease course in Parkinson's disease. And there is experimental model evidence favoring this possibility. So in experimental models, there has been a reduction of alpha-synuclein when using this active immunization. There has been an increase in tyrosine hydroxylase in the striatum, and also a reduction in the alpha-synuclein oligomers. But as always, these are experimental models, and sometimes, or very often, unfortunately, as you all will know, when we try to translate them to clinical trials in human, they are not successful. So this remains to be seen if these approaches will work. The same with passive immunization against alpha-synuclein. So here we use directly antibodies that are infused to the patients to try and block or clear or stop alpha-synuclein pathology. And this has also been backed by experimental models. And in this table from the review paper I was mentioning before, you have a summary of both active and passive approaches of immunization that are under investigation in Parkinson's disease. A few months ago, Profine and Roche released a press release on the preliminary results, on the results of the clinical trial using prasinezumab. And as I am showing this in this slide, the study did not meet the primary objective, but there were signals of a significant effect in pre-specified secondary and exploratory clinical endpoints, and this is now under review by the promoters of the clinical trial in order to define what's the next step. There are also other avenues for trying to handle the alpha-synuclein role in the degenerative cascade. For instance, there could be a misbalance between the tetramers and the monomers of alpha-synuclein in Parkinson's disease, and perhaps strategies that would stabilize these more physiological forms of alpha-synuclein over the pathological ones could be another hypothetical and future approach for treating the disease. And also speaking about alpha-synuclein, but other genes, and this is a figure of one of the first GWAS studies that were published in Parkinson's disease. At that time, only two genes resisted the bonferroni correction, which were alpha-synuclein and the microtubule associated protein tau. But in subsequent genome-wide association studies, several other genes have surfaced as important for the risk of Parkinson's disease, and obviously they could be also potential targets. And LRRK2 gene is one of those genes that have surfaced not only as important in monogenic forms of Parkinson's disease, but also as a risk factor for Parkinson's disease. And here we have another new biotechnological development, which is that of antisense oligonucleotides. So you can use antisense oligonucleotides to prevent messenger RNA to be translated into a protein that you think that is harmful, or that there is a gain of function that is harmful in the pathophysiological process. So there is experimental evidence that this could be also an avenue for treating Parkinson's disease in terms of blocking the translation of messenger RNA of LRRK2. And in this study, there was a reduction when using antisense oligonucleotides against LRRK2. There was a reduction in phosphorylated alpha-synuclein, and there was also a reduction in messenger RNA of LRRK2 and of LRRK2 levels themselves. And obviously, this suggests that even though we still do know quite not that much about the multiple roles of LRRK2 in the pathophysiology of Parkinson's disease, it could still be that LRRK2 has, through its mutations, a gain of function, and then targeting LRRK2 with antisense oligonucleotides could be of interest to develop a new treatment for Parkinson's disease. However, we all are mindful and concerned about the outcome of this, because very recently Roche has made this press release where the study using an antisense oligonucleotide for Huntington's disease, which was tominersen, has been stopped because the independent data monitoring committee found some issues regarding the potential benefit-risk profile. So still, it remains to be seen what's the future for these antisense oligonucleotides in neurodegenerative conditions such as Huntington's disease or Parkinson's disease itself. And there are other pathways. We have the insulin pathway, which in the left-hand side of the slide, you can see a review where the different points of the degenerative cascade where insulin and hyperglycemia could be relevant for Parkinson's disease. And subsequently, a few years ago, the group led by Tom Foltini published this phase 3 clinical trial with a small cohort of patients where they were able to show that exenatide, which is a glucagon receptor agonist that is approved for treating diabetes, was applied in Parkinson's disease patients. And there was a significant difference in the UPDRS favoring exenatide over placebo. And also using dopamine imaging, there was a reduced rate of decline of the DAT scan binding in the exenatide group over the placebo group. We have also the glycocerebrosidase pathway. So here, enzyme supplementation could be an approach, but there could be also some surprising approaches. And a few years ago, a group of researchers suggested that ambroxol, which is an old expectorant, could be a chaperone, helpful for the transit of glycocerebrosidase from the reticulum to the lysosomes. And it is under investigation in clinical trials as an alternative for treating Parkinson's disease as well. And we have iron. I have mentioned before that there is a controversy as to whether iron is an epiphenomenon or is really playing some role in the degeneration cascade. But a few years ago, a group led by David DeVos published this study, which had two legs. One experimental leg where the mice that were pretreated with diferipron resisted the toxicity by MPTP, as you can see in the left-hand side of the slide. And then they had a clinical trial leg where patients who received earlier diferipron, so following an early delayed start paradigm, did better than the patients who received placebo at the beginning, and they were switched later on to the iron chelator. So this led to conduct a multi-center European study, phase 3 clinical trial on conservative iron chelation using diferipron in Parkinson's disease that has finished recruitment and that hopefully will shed some more light on this topic soon. And then we have the approaches that can be more distant in the horizon, but still are a possibility, moving towards personalized medicine. So sometimes we have these negative clinical trials, maybe because we are mixing or combining in clinical trials patients that actually they have the same disease, Parkinson's disease, but maybe every patient has different pathophysiological players or different proportion of the same players. So maybe personalized medicine could be an alternative here. And here we have approaches like the CRISPR-Cas for gene editing that could allow for treating individuals rather than having clinical trials of large cohorts that sometimes can mix patients that are very different the ones from the others, even though when we perform the clinical trials, we try to have samples as homogeneous as possible. And then we have the possibility of modeling disease and identifying new targets also in a very individualized way via the technology of human brain organoids. So nowadays we can create cerebral organoids, and we can have models, as you see in the lower part at the left on the slide, cerebral organoids that are specific from Parkinson's disease patients, and this could lead not only to identifying pathophysiological players or biomarkers, but also to personalized medicine approaches, because we will be looking specifically at cells that are coming from a given patient. And just reaching the end regarding non-motor symptoms, this is another very important area for treating Parkinson's disease, and one of the more important and devastating complications, non-motor complications of Parkinson's disease is cognitive impairment and dementia. And in this review article, in this table, you can see several different approaches that are being under investigation to treat cognitive impairment and dementia in Parkinson's disease. Ambroxol itself, through the ceramide and glucocerebrosidase way, as I was mentioning before, donepezil, for which previous clinical trials have not been favorable. But then we could also here have the possibility of treating Alzheimer type pathology, because this has been shown to be quite frequent in Parkinson's disease dementia in terms of senile plaques and amyloid angiopathy, which are the amyloid beta related pathology, and also in terms of neurofibrillary tangles. So not all, but many patients with Parkinson's disease dementia have these pathologies, and perhaps in the future we could face the possibility of treating our Parkinson's patients not only with anti-synuclein strategies, but also with anti-amyloid or anti-tau pathologies. Obviously, this remains to be seen, and there are several arguments against the amyloid hypothesis in Alzheimer's disease itself. So it is difficult also to know whether it will have a role for treating Parkinson's disease in the future. But here in this review, you see some anti-amyloid antibodies that have potential for near-term approval. One of those that has been more discussed is aducanumab, which has not a low rate of ARIAs, which are these imaging abnormalities that can appear in MRI when treating the patients with anti-amyloid antibodies, which is a concern. But still, these antibodies, not free of controversy, are near of approval or have been discussing for approval, for instance, by the FDA. And who knows if they eventually show usefulness in Alzheimer's disease, whether they could be used for Parkinson's disease dementia. So this is the summary of my talk. This is a table I prepared for the handbook of a Master of Translational Medicine that we offer at the University of Barcelona, and maybe it summarizes what I have been discussing briefly during my talk. So for prodromal Parkinson's disease, maybe we could face in the future the possibility of correcting genetic deficiencies. For clinically established Parkinson's disease, we could have, and also for prodromal cases, we could have these anti-alpha-synuclein strategies or chelators or anti-GBA or insulin or iron targeting strategies. And for advanced PD cases with dementia, maybe we could have other strategies that could be also being developed for other conditions such as Alzheimer's disease. So thank you very much for your attention, and I hope that my talk has been of interest for you.